In this video we will take a look at the slide of liver. This slide is stained with malarial trichrome, whereas the connective tissue is blue, muscle tissue is reddish, and erythrocytes are yellow. The connective tissue from the hilus of liver extends between the liver lobes as an interlobular septa and defines the hepatic lobules. This is the hepatic lobules which surrounding the interlobular septa. The human liver contains less connective tissue than as a liver another animal, for example a pig. So this is in our case. For the better visualization of interlobular septa, we use porcine liver, and the special staining method where the interlobular septa are blue. The interlobular septa contains interlobular branches of the portal vein. It's here. It's also still visible yellow erythrocytes inside the portal vein. The branches of hepatic artery is visible here, here, and here, and interlobular duct, bile duct, which is lined with one layer of cuboidal or columnar cells. And in this big interlobular septa, it's also visible nerve fibers here and here and here, and lymph vessels here and here. So this is one big interlobular septa. For example, here it's a smaller interlobular space with a branch of hepatic artery. Here is a branch of portal vein, and here is a bile duct, and the nerve fibers and lymph vessels. Also, here branch of hepatic artery, bile duct, and branch of portal vein here and here. So all absorbed nutrition from the intestine enters the liver through the hepatic portal vein, and because the venous blood from the digestive organs in the hepatic portal vein is poor in oxygen, a hepatic artery from the aorta supplies liver cells with oxygenated blood, and forming a dual blood supply to the liver. Arterial and venous blood mixed in hepatic sinusoids and flow towards the center of hepatic lobules. And in the center of hepatic lobules is a central vein. For example, here is a center of hepatic lobules, and blood flows from the interlobular space through the sinusoids towards the central vein. Here. Radiating from each central vein towards the lobule periphery are plates of hepatic cells, also called hepatocytes. It's a big cells with pale nucleus, and hepatocytes are arranged into the cords, and between the cords or plates of hepatic cells are sinusoids. This is form of capillaries, very big capillaries, which is lined with endothelial cells with discontinuous basal lamina, and the endothelial cells contains big space, so called fenestrated endothelial cells, for a very quick changes of nutrition between the hepatocytes and blood. So this is the central vein, and the sinusoids. The sinusoids contains yellow erythrocytes, and the hepatocytes have reddish cells in this staining methods. The hepatic sinusoids are separated from the underlying hepatocytes by a subendothelial perisinusoidal space, also called space of disease. Located in this space are the microvilli of hepatocytes and strands of reticular fibers. Unfortunately, reticular fibers are not visible in this staining method. We need another special staining method with silver ions. Sinusoids also contains macrophages called Kupffer cells, derived from monocytes, but in this staining method is also not visible.